We're on, right?
Yeah, we're good. Yeah.
Okay. Thank you, Adriana. And so sharp on the hour, we can get started. Welcome you all back to another fine seminar. Before I introduce our speaker today, to give those of you who may be transitioning from another meeting a couple of minutes, let me just share with you what we did last week, which was definitely a very, very different meeting. So last week, you may remember that we met with some of you to discuss the results of the survey that we had asked you to complete. And in a completely unbiased, properly designed sample of all the people who love us, we learned that 98% of the respondents felt that the rate of the quality of the quality of the fine presentations was between excellent and above average. This is just a little bit of a joke. It was extremely useful. We got like 40 people responding. We had a wonderful conversation with about 20 of you, and the organizers were looking forward to meet. I think we're meeting next week to really try to digest what was discussed and the survey, to evaluate and to really look forward and think forward on possibilities to continue doing what people seem to like and extending and expanding on other possibilities. So thank you all who answered the survey. Thank you all who joined us for a lovely meeting last week. Next week, we're going to hear from my friend and colleague, Martin Kowaleski. Martin is a primatologist, and he's currently working at the Centro de Ecología Aplicada del Litoral, which is one of the institutes of the Argentinian National Council of Research in the province of Corrientes, Argentina. He continues to do research with the black howler population that was the first monkeys ever studied in Argentina going back to the late 70s. So studies of one sort or the other on this population have been going on now close to 40 years. And so I think I was telling Martin yesterday that the way he's imagining the talk will really address some of the suggestions we got from the survey last week, because his research more and more is bringing in a very applied component to what he does. I mean, he's a behavioral ecologist by training, but more and more he's found himself bringing some applied elements to his research in relationship to the conservation in the area. So next week, Martin Kowaleski, I hope you'll be able to join us. So, and today we're going to listen from Mike Webster. Again, I always enjoy making time to refresh my mind on the colleagues who join us, and it was quite a delight to read through Mike's CV and Mike's webpage. Mike told me, and he says so in his page on the Macaulay Library, that he likes to present himself as wearing two hats. The first is that he has been now for I think almost 15 years the director of the Macaulay Library of the Cornell Lab of Ornithology in Ithaca, New York, in the US. And this is a media collection. It isn't just sound like some of us think, but I was learning that over the years they've collected this amazing number of videos and audio recordings and photos from wild birds and more all kinds of animals that they are making available to colleagues and students and educational institutions all over the world. So that's Mike's first hat. The second one is that he now, since 2009, if I remember well, he's been a professor in the Department of Neurobiology and Behavior at Cornell University. And his research has been focused on studying social behavior, sexual selection, animal communication. One of the suggestions in the survey, actually, no, one of the suggestions that we had when we discussed the survey last Tuesday is, what if we had a speaker? What if we had speakers spending a few minutes telling the younger colleagues how you got to where you are? Well, I invite you to check Mike's first couple paragraphs in his website, because it's so engaging. And here you have an undergrad at UC San Diego who thought was going to do marine science, and he ended up taking a course with Sandra Berenkamp and Jack. Oh my goodness, Jack, help me out. I'm blanking. Jack Bradbury.
Bradbury.
So, and that's how he studied animal behavior. And so, for those of us with some years under our arms, you'll be impressed by his advisors, Bradbury and Berenkamp. Then he went on to Cornell for his PhD, and Paul Sherman and Stephen Emlen advised him. And then he did a postdoc at the University of Chicago, where he also stayed for a few years as a lecturer. He had a short stays at Washington State University and soon in New York before landing his position at Cornell. The usual, endless lists of publications, endless lists of grants, of awards, but to say something that I found was different from many others, and I told him so, he has a section in his CV. The title is, with the same font and the same size as many other titles, Student Awards. And it's lovely because it is only about his students, but it gives you in a very nice and engaging way an idea of how many people he's mentored over all these years. Quite impressive, his commitment to undergraduate and graduate education. So it's a pleasure having Mike here with us. He will be presenting today on the topic of, and I want to be fair to the title, sexual signals and divergence in Australian fairywrens, from genes to behavior to speciation. So with that, Mike, please take it from here. Thank you so much for your time. I'm sure we'll have a fantastic presentation.
Okay, thanks very much, Eduardo. That was a very, very kind, generous introduction. I appreciate it very much. And I appreciate the invite to join you all here on this the Zoom screen to share a little bit of the work that we've been doing on Australian fairywrens. And let me get the show going here. It's not letting me optimize for video, and there are some videos in this. Oh, here. Okay, I got it now. I think I need to reshare. 1 second, please. Zoom's always a challenge. Things keep changing.
I know.
Yeah.
We try them. We know it worked. So we should feel fine, Mike, whatever happens.
Okay, so I think you can see now my presentation. Is that right, Eduardo?
No, yet, not yet. We're seeing a beautiful lake. And
okay.
I think it works better if you first put it in presentation mode and then start sharing the screen.
Yeah, I did do that. So let's see what happens now.
There we go.
There we go. Okay.
Yes, now you're set.
Yeah, I'm still not sure if the videos will work in here, but we'll see. Okay. So as I was saying, it's really a pleasure to be here and to share some of this work with you all. I need to first point out, though, that the work that I'm going to be talking about is not by me alone, certainly. It's by really a whole group of different collaborators, students, and friends who've contributed to this work in various ways. And so this screen shows just a few of them. I want to make sure I acknowledge the armies of field techs that we've had working in Australia on this project and helping us collect the data. In part, I do that because I truly appreciate the help and support of all of those field techs, but also in part because Eduardo's son Facundo was one of our field techs, and so we have to I have to make sure I acknowledge that Facundo helped out with all of this. But the work that I'm talking about today, I think, is really highlights the work of three of my collaborators in particular. Dan Baldassari, who was a PhD student working with me and who I see is actually on this Zoom call right now, which is great because Dan can correct anything that I say wrong. So Dan, be ready for that. Emma Gregg, who was a postdoc in my lab when she did the work I'll be talking about today. And then Sarah Khalil, who's currently a postdoc at Cornell working with us. And a lot of the work that we've been doing on Australian fairy wrens, and in particular the species, the redback fairy wren, has revolved around reproductive strategies. Because in this species and some others, males come in a couple of different forms. They're brightly ornamented males that breed and engage in extrapair copulations and those sorts of things. But there are also unornamented males that pair with females and breed also. And so this photo on the right shows an unornamented male with his mate. And so we've been interested in sort of the reproductive strategies of these different types of males, and why they evolve, and some of the hormonal mechanisms underlying them. But that's actually not what I want to talk about today. Today, instead, what I want to talk about is biological diversification or speciation, how it is that a single lineage can split into multiple forms. In this slide, I'm showing a bunch of jumping spiders. And these jumping spiders are a great example of what I'm talking about. There are a lot of closely related species who've diversified in several ways, but the main way that they've diversified, and the main distinguishing feature that contrasts different closely related populations or species, are male sexual traits. So in these spiders, males display to females, face them, and have very elaborate courtship displays where they show off the coloration of their face and some physical other displays. And so Darwin was interested in this process. He called it the mystery of mysteries, how you can go from having a single lineage to two or more lineages. And the process that happens right at that splitting of that lineage into two is a bit of a black box. And he had trouble filling that in. He had some ideas, but it was really later generations of biologists that came along that started filling in some of those boxes. And when they did that, what they started to appreciate is that physical displays and sexual signals play a role in all of this. So this is where I need to ask, can you see the video playing right now? You can, okay, great. Okay, so this is a male jumping spider displaying to a female. He's showing off his face. He has stridulating calls, so some acoustic features. He has a physical display where he raises and he snaps his forelimbs. And all of this is the way that a male jumping spider woos his mate, tries to convince that female that she would like to mate with him, right? I'm going to jump up a little bit in this courtship. The male is getting a little more excited here. The female is not leaving, which in spiders, so the female's not leaving and she's not trying to eat him. And in spiders, that's a good sign of female receptivity, right? So he's very happy that she's not leaving. He's getting more excited. And now he's going to try to mount her and mate, but I have to stop the video at that point because Eduardo told me this has to be a PG presentation. And actually what you're missing here is a very disappointed male jumping spider, because he's about to discover that the female he's been courting is actually freeze-dried, and so unable to copulate, which is why she was not leaving. So this observation that male courtship signals can vary between very closely related species really prompted Ernst Mayr and others to develop this idea of the classic model of speciation, the allopatric speciation model. And the basic idea is that one population gets split into two populations, and then those populations diverge genetically, so that if they come back together, hybridization between those two populations is costly because there's been too much genetic divergence. And so hybridization is selected against. And that leads to the evolution of these reproductive signals, these sexual signals that males and females use to court each other. The idea being that those signals diverge so that hybridization does not happen, right? So that's the classic model of speciation developed by Mayr and others. But more recently, not super recently, but a few years later, alternative models came forward. And the most important one was what I'll call the sexual selection model, developed by Mary Jane West Eberhard and others, Ruth Landy. And in that model, there's still allopatric separation of a single population. So one population gets split into two, and there's little or no gene flow between those populations. And things diverge, but in this model, the thing that diverges first are those sexual signals. And so the idea is that the sexual signals diverge from each other so that if those populations come back together, they're not interested in mating with each other because there's been too much divergence in the signals, and you have reproductive isolation, even though if those individuals from the two different populations did interbreed, their hybrid offspring would be perfectly viable. So that's the alternative sexual selection model. And the big difference between these two models is where divergence in sexual signals occurs. In the classic model, it emerges very late in the game, whereas in the sexual selection model, it's one of the first steps where we have divergence between populations in those sexual signals, and that's what's leading to reproductive isolation. So this model's been out there for a long time. There's actually been some really interesting work done in recent years to help support the idea of the sexual selection model, including work on jumping spiders. That's why I highlighted them. Some nice work by Carrie Shaw and some of her colleagues on Hawaiian crickets. And then some really cool work recently on different birds. One project being Sheila Turbeck looking at the seed eaters in South America, and all of these studies are showing what's emerging as a common pattern, which is that in populations that are closely related, there can be strong divergence in sexual signals, but little or no other genetic divergence, which implies that if the populations were to interbreed, they would be capable of doing so, but they don't interbreed because these sexual signals have diverged. Right. And so that's interesting. But as a behaviorist, I'm interested in, well, what are the behavioral mechanisms underlying there? How do populations diverge? How do mating preferences diverge or not? And what are the behavioral changes that are occurring that lead to the reproductive isolation beyond just a change in sexual signals? So that's what we've been interested in looking at. We've been interested in this clade of fairywrens in Australia, where the species are relatively closely related. They're in a single clade. But what differentiates these different species from each other is male plumage coloration. So you can see in this figure here, males look very different from each other from one species to another. But in all of the species, females are sort of a cryptic brown. So females look very similar. And what is really distinguishing is male sexual signals, in particular plumage color. And we've been studying several of the birds in this group, but we're focusing in particular on the redback fairy wren, the one at the farthest left, which is Melurus melanocephalus. And the reason we've been focusing on that species is there's a lot of aspects of its biology that lend itself really nicely to a study of speciation and divergence in signals. So it's the redback fairy wren is an Australian songbird, a passerine. It's socially monogamous, so one male and one female pair with each other. But there's cooperative breeding. Sons often stay at home and help mom and dad raise more offspring. And even though they're socially monogamous, there's a very high rate of extrapair paternity. So if you genotype the offspring and compare them to parents, you find that about half or more of the offspring are actually sired by a male other than the social father that's feeding them at the nest. And so this high level of extrapair mating, extrapair paternity, opens the door for strong sexual selection. Oops. But what's really particularly important is that plumage color is important to mating. We've done a lot of different experiments showing this, that females are really paying attention to coloration when they're choosing mates. I'll show some of those data in a few minutes. But the plumage color has diverged across populations. And I'll show that also in a minute. So actually, I feel like I'm missing a slide here. Oh, no, we're good. Okay, so the research questions that we wanted to tackle in this project were all related to this idea that sexual signals diverge between populations, and that leads to reproductive isolation. So we're interested in the question of what causes divergence in these signals. Is it ecological or drift or sexual selection? We've asked the question, do these divergence in signals lead to reproductive isolation between populations? So if they diverge in signals, are they less likely to mate with each other? We're interested in the behavioral responses to those signals because mating depends on those behavioral responses. So we wanted to know, how do females and other males respond to the signals? And then for reasons that I'll get into later, we are also interested in looking at the genes that are involved in producing these different kinds of signals, right? And so we studied this in the Australian fairywrens. It's a handy species because of what I said before. There are two recognized subspecies of the redback fairywren, and the main thing that distinguishes them is male plumage coloration. So across northern Australia, there's the subspecies Cruentatus, which I'm going to call the red population, a red subspecies, because males in this subspecies have a crimson red back, a deep red back coloration. But along the East Coast, in this same species, is a different subspecies, Melanocephalus, which I'm going to call the orange subspecies of the orange population, because male plumage coloration in this species, males have an orange back rather than a deep red back. And that is the main feature that distinguishes these two subspecies, is whether the male has a deep red back or an orange back. And Scott Edwards and Yoon Lee, his graduate students, showed several years ago that these two subspecies are genetically distinct, and they probably diverged during the Pleistocene. So they've been separated for several 100,000 years at this point. And the field work that we've done has actually occurred at 3 different locations. We've done a lot of our work in Herberton, which is actually in the middle of a contact zone between the two subspecies. We've done a lot of work in Brisbane, in the southeast of Australia, which is right in the within the range of the orange subspecies. And then we've done some work at Darwin, which is within the range of the red subspecies. So we're looking at populations that are red in male back coloration, populations that are orange in male back coloration, and then populations that are a mixture of the two. So one of the first things we did was a transect across Australia, collecting blood samples and feather samples from across the entire transect to look at male plumage coloration and also to look at genetic differentiation between these populations. And so we collected these feathers from the different populations. These numbers in this map are the number of samples we collected. And you can see just from this photo that there's red and orange feathers, just as I showed in the previous photos. And if you look at the change in plumage coloration across this transect that we ran, you see that the y-axis on this graph is male hue, or the color of the back, where high values are red and low values are orange. And you can see as you go from east to west, that's the x-axis, is distance, geographic distance. You go from males who have very red backs until you get to about the 2,000 kilometer mark, and all of a sudden they switch over very quickly in space to having orange backs. So there's a steep climb in male back coloration along this transect. You have red males, then a decline, and then orange males. And the center of that is near a town called Cardwell, which is unimportant. And so this dotted line basically shows the middle of that transition between plumage coloration between red and black. So I won't get into it now, but Dan, who's online right now, did a lot of analyses to show that this divergence in back coloration is not due to any ecological differences between the different populations, but rather due to random drift and probably sexual selection favoring different colors. And so it's not an ecological divergence, but a drift. And so sexual selection drift is causing the change in back coloration. I'm happy to talk about that later if you'd like, but to keep this talk manageable length, I cut that part out. But the big question I want to focus on here is, does the difference in back coloration lead to reproductive isolation between the different populations? And the way we tackled this question was by looking at the genetic line, so the differences in genotype across space, and compared that to the differences in back coloration. And the main prediction is that if back coloration, plumage coloration leads to reproductive isolation, then the plumage cline and the genetic cline should be concordant. They should match each other. And so we tested that, and I should say, Dan in particular tested this by taking a genotype by sequencing approach, and he isolated many SNPs, single nucleotide polymorphisms. So these are genetic loci that differ between different individuals. So they're polymorphic. End up looking at about 645 of those after filtering through all of the SNPs that he found. And he plotted the frequency of those SNPs across space. And I'm showing about 9 clines here. And you can see for each one of these genetic loci, one allele is fixed at one end of the distribution as you move through space. And then you hit a point where it transitions so that the other allele becomes fixed in the other population. So the frequency of each genetic marker is changing over space, where it goes from high at one end of the transect to low at the other end of the transect, just as plumage color changes along the transect. So then Dan compared that genetic cline with all of those single nucleotide polymorphisms. He compared that cline to the plumage cline in coloration, plumage coloration. And so here again, this is the cline in plumage coloration, how the change in plumage color occurs across space, going from east to west. And here's the genetic line. The gray lines are individual loci, individual SNPs, and the black line is the average. So it's basically a hybrid index. And you can see there's a genetic cline and there's a plumage cline. But the average genetic cline is shifting several hundred kilometers to the to the west of the plumage cline. So the two clines are not concordant with each other. They're significantly different from each other. And that was a surprise. We were surprised to find that these two did not line up with each other. And what this strongly implies is that that the plumage color is not leading to reproductive isolation, but rather that plumage color is introgressing. It's moving across the genetic line from one subspecies to the other. And so the genetic break between the two subpopulations is this other dotted line. And so there's this big transect or big part of Australia in the middle there around Cape York Peninsula where males have one plumage type. They have red plumage, but they have the orange genomic background. And this is just showing that same thing in another way with a population structure plot. And so you can see the red bars, each one of these bars is an individual bird. Red indicates an amount of their genome that is assigned to the red subspecies genome. Orange is the amount of their genome that is assigned to the orange subspecies genome. And there's a genetic break and a plumage break. And basically what's happening here is the red, the plumage coloration is moving from one subspecies into the other. The red plumage is moving into the orange genomic background. So plumage is not leading to reproductive isolation, but quite the opposite. We see introgression of plumage from one subspecies to the other. And this is actually a pattern that's starting to emerge for a whole bunch of different subspecies of birds where people are starting to look genomically. Plumage variation and differences in plumage coloration do not line up well with genomic differentiation between subspecies. So there's a lot of change and movement and dynamics to plumage coloration across the background of a genomic differentiation. But that got us interested in song. We were interested in like, wow, well, what about song? These birds use plumage to attract mates and to repel rivals, but they also sing, right? And so do we see the exact same thing with song? And that's where Emma Gregg comes in. Emma was a postdoc in the lab. And so Emma went to the Macaulay Library, and she also visited many populations in Australia and recorded birds in all of these different populations. And she compared their songs to each other. And this is what they sound like. So here's the red subspecies song. So kind of a high twittery sort of song. Here's the orange subspecies song, which is also twittery, but it's a little bit lower pitched. It has, it's longer, or sorry, shorter. It has some introductory notes. So there are differences between the songs of these two subspecies. Even though to humans, they sound kind of similar. And Emma measured a whole bunch of things about these songs and quantified the differences. And what she found is that the blue dots in this, these are just three principal components that summarize the measurements, song measurements she made. And you can see that the blue populations, which is the populations in the red part of the distribution of the red subspecies distribution, are differentiated from populations that are shown in orange and yellow here. The orange populations are populations that are genomically and plumage-wise orange subspecies, but the yellow populations are species where males have the genome of the orange subspecies, but the plumage of the red subspecies. And so what this means is that we have in the far north populations of these birds that have the genome and plumage of the red subspecies. In the far east of Australia, we have birds where males have the plumage and the genome of the orange subspecies. But in between, we have populations where they are chimeras. Males have the plumage of the red subspecies, but the genome of the orange subspecies. And they also have the song of that orange subspecies. So the genetic cline and the song cline match up perfectly, whereas the plumage cline is shifted significantly to the east. So this strongly suggests that song is a reproductive isolating mechanism, even though plumage is not. So song does seem to keep these populations somewhat separated, and that's probably why they continue to be genetically differentiated. But plumage is introgressing from one subspecies into the next. So that brings up the question: well, why? Why would plumage introgress from one subspecies to the other, but not song? And so to understand that, we realized we needed to look not just at the signals themselves, but at the responses to those signals. So we did a couple of experiments to try and get at these different hypotheses. So why would plumage introgress but not song? One hypothesis is that it might have to do with the mode of transmission. I'm not going to get into that hypothesis right now. I'm happy to talk about it later. But the two hypotheses that I want to do want to talk about is that male-male introgression limits song introgression. So another way of saying that is males use song to compete with each other. And so that competition restricts the movement of song across subspecies boundaries. And the alternative, or the other hypothesis, it's not an alternative, they can both be true, is that females prefer red males. And so female preferences may be driving the movement of the red plumage from the red subspecies into the orange subspecies, right? And so Dan and Emma together did this really massive experiment that required several hundred replicates doing playbacks to different birds in different populations. And they did this experiment both in Darwin, which is right in the center of the red subspecies population or distribution. And they did these experiments in Brisbane, which is in the center of the orange subspecies distribution. And so they're looking at the responses of males who are red and males who are orange to their different treatments. And what they did is a massive mix and match experiment where they could do a playback of a song from a speaker underneath a mount of a realistic looking male, right? So they had a fake male with whatever plumage color they chose, and underneath that, a speaker that was playing whatever song they chose. And so they did all possible combinations. They did local plumage coloration and local song coloration. They did local plumage coloration, but non-local plumage coloration. So they could do orange plumage, red song, red plumage, orange song, all the different permutations. And they had heterospecific controls as well, where they would have a different species song or different species plumage, and they could do these different playbacks, these different replicas, to look at whether males are responding mostly to plumage color or mostly to song. And in the paper, we said that we use lifelike models. These are the lifelike models. They are modeling clay that are kind of vaguely formed in the shape of a bird. But the feathers on the outside are real feathers, and in particular, the feathers on the back are fairy wren feathers. So they use actual fairy wren feathers for their experiment, but they could swap out either using red or orange feathers on the back. So like I said, they did several hundred different trials to disentangle plumage and song responses to plumage and responses to song. And they scored the responses of males. So they did these playbacks on the territories of males, and they looked at the responses of those males. So what I want to do now is, rather than show you a bunch of graphs, I'm just going to show a video that I think really sums up nicely the results of this experiment. So in this video, this is a playback at the Brisbane population. So this is a population where males have an orange back. And in this particular treatment, the model has a red back, right? So this is a non-local male plumage color. But the song that's being played underneath this male is the orange population song. So it's the local song, but non-local plumage coloration. And this is what they saw. So that's the orange song being played. And here comes the local male. You can see he has an orange back. And he attacks the model pretty vigorously because of the song he heard. He doesn't care at all that male has a red back. He attacked the model because of the song. And here's another trial where they used the mount as a heterospecific control. It doesn't even look like the same species. And the territorial male is attacking that bird because he's hearing the local song. And so the upshot of that experiment that Dan and Emma did was that males really rely on song to make decisions about whether or not to attack another bird that's singing, but they don't pay that much attention to plumage coloration except in sort of very local interactions. Once the male comes in, plumage color has a moderate effect on whether he actually attacks the model or not. And that's what these graphs show. I'm going to skip over those for time. But basically, the results of that experiment show that male-male introgression or male-male competition is based a lot on song, and that's probably limiting the introgression of song from one subspecies into the other. But it permits, because males are ignoring plumage coloration for the most part, it could potentially permit the introgression of plumage coloration. Males don't care so much about the plumage coloration. So if some other force is pushing plumage from one subspecies to the other, males are not going to, male-male competition will not interfere with that. So the question then is, well, what's driving that introgression? Is there some force driving it? And our hypothesis is it was female mating preferences, that females actually prefer red over orange, no matter what population they live in. And so a form of a pre-existing mating preference. And to test that, Dan did an experiment in Brisbane in the orange population where and I did want to say that the reason we thought plumage coloration is important is males display that back coloration to females during courtship. They puff up their backs in something we call the puff back display. And then they also display flower petals to females that usually match the back coloration. So redback male fairy wrens use red petals when they're courting females, and that coloration matches the back. So we thought ahead of time that females probably cared about plumage coloration. And so Dan did this experiment in Brisbane where males are colored orange, where he actually changed the color of some of those males with magic markers. So he would manipulate the coloration of the male's back. This is a population where males are normally orange, and so he used red markers to make some of those males orange. And then he used clear markers to sham operate some of those males. So he would take a marker that was clear in coloration. I don't know why, but artists use clear markers for something. And so you can do the same manipulation of the males, but not actually change his back coloration. And so you can see here, going from left to right, the controls are males who were captured but not manipulated in any way with the marker. The colored pre are the color of the orange males before their color was manipulated. The sham pre are the males when they were first captured before they were their color was manipulated. The sham post are males who were marked with the clear marker, and you can see that that did not change the coloration of their back at all. They're still orange. But the colored posts are males who were manipulated with a red marker. And you can see the red marker did make them much redder than the orange males. And in fact, the rightmost bar there is the coloration of the other subspecies, the red subspecies. So Dan was able to very closely mimic the coloration of the other subspecies with his markers. And then he just let those birds go, and he let them go about their business and do their nesting and everything like that. He found all of the nests. He collected blood samples from the nestlings, and then he genotyped the nestlings and the adults so that he could compare those nestlings to the adults and basically ask the question, is the father at the nest the sire of those offspring? And so he could quantify reproductive success of males in all of these different treatment categories. And so if you look at within pair success, so this is the number of young that a male produces in his own nests, there was no real difference between the different treatment groups. Control males, sham operated males, and colored males, the treatment males, all sired, on average about one nestling in their own nest. But if you look at extrapair young, so the number of young that males are siring in the nests of other birds, other males, there was a big difference. So males who were reddened, males who had their back plumage changed to become more red, sired many more extrapair offspring than did either controls or sham operated. And again, this is in a population with orange males. So females in this population are showing a preference for males who have plumage of the other subspecies. They prefer males with redder plumage. So if you look at the total reproductive success add up within pair and extrapair, you see that the reddened males are doing quite well. And so what this basically mimics, this experiment mimics a situation where a mutant male with redder plumage emerges in a population, or an immigrant male with red plumage moves into the population, and it shows that red male would have very high reproductive success and thereby spread whatever genes he brings into the population with him. Right? So the upshot of both those experiments is that male-male interactions, male-male competition limits the introgression of song, but permits the introgression of plumage color. And in sexual selection, female mating preferences actually promote the introgression of red plumage into orange populations. Right. So now for the last few minutes, I just want to talk about some very recent work we've been doing, trying to isolate and look at the genes that might be responsible for this plumage coloration. And the reason we wanted to do this, one of the reasons, is that all the behavioral work we have suggests that these sexual signals, in particular the plumage signals and the responses to those plumage signals, are not leading to reproductive isolation, but are actually facilitating introgression of phenotypic traits from one population to the other. And we wanted to ask the question, do we actually see that at the genetic level? Can we show that genes are being driven by sexual selection from one subspecies into another? And to do that, we needed to find those genes. And so again, the question we're asking here is selection, in particular female mating preferences, driving the introgression of specific alleles at loci that affect plumage coloration. And so we're predicting for this that there are loci that affect plumage coloration, that it's not an environmental trait, but that it has some genetic underpinnings, that those loci will track the decline in plumage coloration, and that we'll see signatures of positive selection on those loci. And this is work that's being done now, or just recently being completed by Sarah Khalil, a postdoc here at Cornell. And to think about the genetic underpinnings, I think it's really important that we understand, well, what is it that makes a male's back red in the first place? Because it's not that simple. The coloration is coming from carotenoids. Animals cannot synthesize carotenoids, but they can modify carotenoids. And so that means that any carotenoids that are being deposited in the plumage, the feathers of the male, have to come from his diet. So birds are uptaking carotenoids through their diet. But a lot of the carotenoids that are out there in nature that are in the insects and things that birds are consuming are actually carotenoids that code for yellow rather than red. And so these carotenoids need to be secondarily modified to give the redder hues, the redder coloration. And that is thought to happen mostly in the liver. And so in the liver, there's secondary modification, enzymatic modification of these carotenoids, and then those carotenoids are transported from the liver to the growing feathers where they're deposited. And so anywhere along this route, you could have genes that affect that process. And some of Sarah's earlier work showed that indeed there are genes that are regulated by testosterone that are important to modification and transport of carotenoids. And so for her more recent work, she's been asking the question, do some of these genes differ between the subspecies? And do we have evidence of introgression of some of those loci? So to do this, she took a genomic approach. She went to the hybrid zone. This is an area where we have orange males and red males and all mixture in between. And she collected 20 different males or blood samples from 20 different males, as well as feather samples. And she chose the 10 reddest males she could find in her population in the hybrid zone and the 10 orangest males. So she's choosing males from the same population who differ in back coloration. And then she also looked at the allopatric population. So she went near Darwin and collected males who have red plumage and collected blood samples from those males. And she went down to Brisbane and collected blood samples and feathers from males in that population where all males have an orange orange backs. And she did whole genome sequencing and resequencing, and she aligned all of her sequences to a reference genome for this subspecies that we've developed. And her average coverage was about 10x, and she found 28 million SNPs in that sequencing. And so if you just look, you throw all that genetic information into a principal components analysis to say, are these populations differentiated from each other? You can see that the orange and the red populations, that's the Brisbane and the Darwin populations, are quite differentiated from each other, not surprisingly. But in the hybrid zone, the birds are all very genetically similar, regardless of the color of the male's back. And we expect this. This is an interbreeding population where male sexual signals vary across individuals, but otherwise they're interbreeding. So we expect a lot of genetic homogeneity within that population. So then she produced Manhattan plots where she looked for differences, genetic differences along the entire genome of the different birds that she looked at. And so first she looked at comparisons between the two allopatric populations, so between the red population and the orange population. And she found that overall genetic differentiation was actually pretty high between these allopatric populations. Again, not too surprising. They've been separated since the Pleistocene. But there are a lot of different peaks that go above the average differentiation between the populations. So these are peaks and genomic sequences that are particularly differentiated between the two subspecies. Some of those might code for coloration of the back of that, but a lot of them probably code for many other things. So to really try and get at which of these genomic regions might affect back color, she also looked within the hybrid zone and compared the red males to the orange males and found a much lower level of overall differentiation, again, because it's an interbreeding population, but still found some peaks where orange males were quite different from red males. And a lot of those peaks lined up with the same peaks she found in comparing the allopatric populations. So these are parts of the genome that may well contain loci that affect back coloration. And she honed in on those. And in fact, she made what are called amplicons, so short 300 base pair segments of DNA that were based on about 120 different segments of the genome that are different between the two subspecies. So she chose loci that are differentiated between the two subspecies, and she amplified those amplicons in all of the individuals across all 16 populations. So she looked at the genotype at these differentiated loci in 285 different individuals. And she asked which of those loci might line up with the decline in plumage coloration. So again, here's the work from the previous result from Dan's work. We have the black line with the gray air bar around it is the genetic line. So that's a genetic differentiation between subspecies. And that red curve is the plumage coloration difference between the two subspecies. And onto this plot, Sarah added all of the different loci that she looked at. And what she found is that many of them, about half of them, lined up with the genetic line, which is what we'd expect. These are probably loci that are differentiated between the two subspecies, but have nothing to do with plumage coloration. In contrast, she did find a handful of loci that line up very nicely with the plumage line. And so here's one of those. This is fasen. It's an enzyme that's it's involved in fatty acid transport. Carotenoids are basically fatty acids. And you can see this fasen line, that's the blue line, lines up almost exactly with the plumage coloration line and is shifted well to the east of the neutral genetic line. And so this is what we expect to see in any locus that affects plumage coloration and is being transported or introgressed from one population to the other. And then she looked at the genotypes of different birds at this one locus. She found that the genotype of an individual bird, whether it was homozygous, heterozygous, or homozygous for the opposite allele, lined up very nicely with the male's plumage coloration. So strong evidence that this locus affects plumage coloration and that it's introgressing from the red population into the orange population. And she found a handful of other loci like that too. So here are 4 of them here, where each locus, the genotype at that locus is associated with male back coloration, and the frequency of that locus, the decline in the frequency of that locus, matches up very closely with the decline in plumage coloration. So it looks like what Sarah's been able to identify here and show is that female mating preferences that are driving the introgression of male plumage coloration from one subspecies to the other are also driving individual alleles at specific loci that affect plumage coloration. And when she looked at where those alleles were located in her genomic analysis, she found, for example, that fasen, the gene I was just highlighting, is on scaffold 453, which is exactly one of those peaks that she sees between the allopatric and plumage color comparisons. And when she looks at, I'm not going to explain these figures in any detail because I'm running out of time. Basically, when she looks at genetic differentiation in that scaffold right around that gene, fasen, she finds that genomic, there's suppressed variation, genomic variation around that gene, and Tajima's D, which is something that indicates selection, goes crazy around that gene, and so very strongly showing signs of positive selection in that area of the genome that selects for plumage coloration. So the lessons from the redback fairy wren that we've gotten from all of this work are, first, that back coloration diverged not because of ecology, but because of drift and sexual selection. That plumage coloration does not prevent interbreeding, but song does. That allows the introgression from one subspecies to another of genes that likely affect plumage coloration. And probably a lot of this is driven by pre-existing female mating preferences, where females, regardless of what population they're in, have a mating preference for males with red plumage coloration rather than orange. So more general conclusions for like speciation process as a whole, moving out back away from our subspecies. Sexual signals, I think this work shows that sexual signals can diverge via drift and sexual selection. That divergence may not lead to reproductive isolation. And what really is important is how birds respond or individuals respond to those sexual signals. So we need to look at the response to signals as well as the divergence of signals. And that different signals and modalities may have different effects on reproductive isolation, even within the same system. And the final take-home message for me is that speciation is a very messy process. There's a whole lot going on at that initial divergence between populations. A lot of that messiness is driven by differential responses to different kinds of sexual signals. And with that, I want to thank all of the people who were involved with this research, different institutions. I really, in particular, want to give a shout out to Emma Gregg, who was a postdoc and who tragically passed away just a few months ago. Emma was a very good friend and collaborator, and none of this work would have been possible without her help. And with that, I'm happy to take any questions.
Thank you, Mike, very much. Very, very interesting talk that connects to so many of the presentations we've had in the past. So I'm sure there will be tons of questions from the audience. Just a quick reminder. The first thing is, since some of you may leave, remember next week we have Martin Kowaleski, primatologist from Argentina, discussing behavioral ecology and population biology of black and golden howler monkeys. You know that most of you know our routine from having a look at our audience today. I invite you to type a question mark in the chatting window. If you're a student, postdoc, or classify yourself as a young colleague, and by doing that you get priority, just type an S or something besides the question mark, and we'll jump to give you priority. So just start typing question marks in the chatting window, and we get Mike to engage in a lovely discussion as we always have. So I see, okay, our first question. Oh, please, one more time, since maybe Mike hasn't met many of you, if you can please say where you're joining us from and what's your affiliation. So our first question comes from Clara Jones. Clara?
Hi, thanks for the talk. I'm Clara B. Jones, social evolution, retired. I just quickly want to say that I studied with Jack also in the 70s. Can you hear me?
Yes, I can.
Okay. Yeah, and of course, that was a life-changing experience. Secondly, I just wanted to say, especially to the females in the audience, that Maria Servadio is at Duke or University of North Carolina, one or the other, is probably the female who's worked on this topic longest. And I just wanted to highlight her. I don't know this literature. This is my question. But when I was in graduate school in the 70s, ages ago, of course, there was a wide and vocal consensus that speciation could not be caused by sexual selection. And I've been wondering throughout your talk and otherwise whether or not that consensus has changed, and how you got to the point, if it has changed, of convincing people that it was possible?
Yeah, no, that's a great question. I would agree with you that that was the consensus, and the main argument back then was that if you just have a little bit of hybridization, so if the reproductive isolation caused by sexual signals is not very, very strong, you have a little bit of hybridization, because the offspring are viable, that would lead to dissolution of the two subspecies, and they would meld back into one. And that's definitely something that's seen a lot. That's seen in our system. So in our system, in the hybrid zone, there is absolute interbreeding between birds that have different plumage coloration. It's difficult, though, for a male singing the wrong song to establish a territory there. And so I think that's why we see song playing some reproductive isolation, but not plumage. And there, I would say in very recent years, there's been a lot of convincing evidence that sexual signals by themselves can lead to reproductive isolation, even if hybridization leads to viable offspring. How common that is, is that just in some isolated systems, or is it a very general pattern? I think is still open for debate. I actually think it's pretty common, but it's very messy. There's definitely cases where song can lead to isolation, but not plumage. So it's going to be highly variable across systems. It probably depends on how long the populations have been isolated and how divergent the signals are.
Thank you. Good luck.
Excellent question.
Karsten, I'm going to skip you. We'll go first to Heather Kenny, who introduced herself as a student or postdoc. Heather, please, it's yours. If you can tell us where you're joining us from and your affiliation.
Thanks. Yeah. I'm Heather Kenny Dudella. I'm a grad student at the University of Colorado in Boulder. I'm working with Becca Safran, thinking about sexual selection in barn swallows. And
awesome.
Mike, I think we got to meet at the AOS meeting that was here.
We did, yeah.
In Colorado in October. Yeah. So yeah, I really enjoyed your talk. And I was wondering if you have any idea, like what was the original basal plumage state? Like, was it more red or more orange? And then which one changed more?
Right. The patterns that we see are most consistent with the idea that the basal ancestral coloration of males was more orange. And then during the Pleistocene, when the two populations were separated, in one population, we saw mutations that led to redder plumage coloration. And so when they come back together, females see these redder males and they had a preference for them. And so that pre-existing preference is what's driving the introgression. I think some of the genomic evidence we have that from Sarah's work supports that, but there's also some confusing results. In particular, even though we see strong positive selection on alleles that are affecting plumage coloration, it seems like the strongest signal of positive selection is on orange alleles. And so we expected to see the opposite. So we're still trying to sort out some of that, sort of the genomic evidence. But our hypothesis is that orange was the ancestral state.
Cool. And I was also curious if how much variation there is within the orange population across males, if some males are more on the red side, and if you can actually see that there are reproductive benefits of that within just the orange population.
So we tried to look at that, and in particular Dan did, so he can chime in too. But within the orange population, there's not a lot of variation. Around Brisbane they're very orange. Around Darwin they're very red. But in the hybrid zone or the mixing zone where the two come together, you have the full spectrum from red to orange. And in that population, we haven't published this yet, but in that population where you have a large variation in male plumage coloration, we do see redder males having higher extrapair mating success, which is consistent with Dan's experiment.
Nice.
Karsten, you're next.
Yes, I'm Carsten Schradin, working at the University in Strasbourg, France, and in South Africa on mammalian social evolution. Thanks for a great talk, Mike. And you mainly already answered my question. So it seems these two populations have been separated at some stage. On the, what is it, East Coast, you have females that prefer orange song but red males. And now it seems that the red males with the orange song from this, what is it, north of Queensland are invading this population and spreading their genes more and more. Is that what you're describing?
Yes, the female, we don't know anything about female preferences for song. We know how males react to song, and we can make inferences about how females may respond to song. But we do know how females respond to back coloration. We know how males respond to song, and the male response to song would make it difficult for a male singing the wrong song to establish a territory. But because males don't pay as much attention to plumage coloration, it allows for the introgression of male color, redder males into orange populations.
Yeah, thank you.
Sure.
Amy, you're next.
Hello, I'm Emmy from the Max Planck Institute in Seewiesen, close to Munich in Germany, and I worked with blue-tits. I have a question about what regarding again the female preferences and what exactly prevents the spread of the red species. Sorry about that. So you mentioned the extrapair success is evidence for the female preference, but that's relatively indirect. It could also be due to male effects. And
Sure.
then also, if you consider the red males with the red song, you'd think they're optimally suited to obtain maximal success as soon as they disperse a little bit into that orange area. What prevents that this rapidly sort of happens? Is it because males don't disperse that much?
Yeah, so taking the first question, you're absolutely right that the results of that experiment by Dan with coloring the back doesn't rule out male-male responses to back coloration. And males do, we know from the other experiment, the males do pay some attention to back coloration, but it's a much more muted response compared to how they respond to song. But we do have other lines of evidence that females pay attention to back coloration. As I said, if you look at the male displays during, especially extrapair courtship, males are displaying their back, they puff their back feathers up and display it to the female. And males are jumping around females during the whole display. And Joe Welkin, who is another graduate student in the lab, did some high-speed videography of that display. And when you slow it down and look at the display, what you see is that the male, when he jumps over the female, is actually doing a somersault. So the whole time that he's jumping over her, he's pointing his back at her. And so that it's like he really wants that female to see his back. And so that's, again, it's observational. It's not an experiment, but it's consistent. And then we have done experiments in the mate choice experiments in the lab, giving females a choice between different kinds of males. And we know that females prefer colorful males over brown males. We have not done, and so we know that females pay attention to coloration in that sense. We've not done the choice experiment where we give a female choice between a red and an orange male. That would be an obvious thing to do. We just have not done that yet. And then your second, males who sing. The answer to that is that dispersal is very limited in these birds. They do not move very far. Males are dispersing maybe 2 territories away from where they're born. And in many cases, they're inheriting the territory where they're born. Females disperse further than males. So because females can take the genes of their father, but they're not necessarily taking his song to the next population if they're dispersing, that allows for the movement of plumage genes, but not song, which is learned.
Great, thanks. Very, very
Sure.
interesting.
Thank you.
I think I come next. Eduardo Fernandez Duque, Yale University. Mike, when we met earlier, we were talking about the challenges that some of our systems pose to really get at the neurobiology of all of this. What can be done to assess perception? Because at the end of the day, I was quite struck by the idea that this bird doesn't recognize. I mean, those models, those models that they placed there and really confused the male, attacking what didn't look like a bird to any of us. I mean, it's like I assume that birds are these extremely visual guiding animals, and now you have a poorly made bird that looks a little bit like, and they attack it. So it got me thinking about perception. What can you do to really measure that? Because as you get more sophisticated in assessing response, etc., what matters is how animals perceive the stimuli you're quantifying.
Right.
Are
Yeah.
you guys doing anything? Has anything been done in the lab or?
No, we're not. I mean, their perception occurs at many levels, right? And so there's the initial perception at the level of what the eye or the ear or whatever that's detecting the signal. But then there's higher order processing that brings it all together. And presumably the higher order processing is what you're getting at. And how can they be fooled by something that doesn't look that much like a bird, but just has the right color, right? And so we have not thought that much about doing that sort of thing. One thing we are very interested in doing, we haven't started doing it yet, but we've talked about doing it, is looking more at the level of the eye. I would really like to look at the retinas of these birds and compare closely related species, and also the red versus orange populations, and look at the photoreceptors in their eyes and how those are populated in species like the redback fairy wren, where you expect a lot of red photoreceptors compared to its closest relative, males have blue and white plumage, and so you'd expect very different photoreceptor distribution in the retinas of those eyes. And then within the redback fairy wren, comparing red populations to orange populations, my hypothesis is that they'll be the same because females seem to really like red. I think that at the photoreceptor level, they just are well built to see red. But beyond the eye, if you get deeper into the system, we haven't really toyed with the idea of what to do with that. It'd be a fascinating system. I think you need a model system, and fairy wrens, as fun and cool as they are, they're not a model system for doing neurobiology.
Thank you. So Zuleyma, if you can bear with us a minute, we have a question from our colleagues joining us via YouTube. Adriana, do you want to bring that up?
Yes, I will. So question from YouTube comes from Nina Trufas, and it says, hello, thanks a lot for your very interesting talk. Do you have hypotheses regarding why would females prefer the red plumage over the orange one?
Yeah, we have ideas that we haven't tested yet, but there's been a lot of hypothesizing by many people, I guess probably most prominently Jeff Hill, but also others, that carotenoids are good indicators of health. They're difficult to obtain from the environment, so they should be good indicators of good genes, basically male condition. And so you could presume under those sorts of ideas that female preferences for redder males would evolve and be adaptive, right? So females, it may be an adaptive preference for redder males, but male signals then are responding to that preference, and that's why when males who have some sort of mutation that leads to redder plumage from orange plumage, that those alleles at those loci could be favored strongly by the female mating preference. But the origin of the female mating preference for redder males, we can only hypothesize at this point. And it would not surprise me at all if it has something to do with some sort of good genes argument. But we've never tested that.
Thank you. I apologize, Karsten, you're next. I forgot about you, dude. And then, so we'll do Karsten, then Zuleyma, and then Adriana.
I asked my question. Sorry, it was so boring. I forgot it, Eduardo, but you can go to the next.
Well,
it was a good
I'm
question.
getting confused. I must have, okay, I apologize. I told you guys I can multitask. So
It's Zuleyma. It goes to
we're going with Zuleyma.
Okay, hi, Zuleyma Tang Martinez. I'm retired from the University of Missouri St. Louis, and my interest was primarily in issues having to do with social behavior broadly, chemical communication more specifically, as well as other areas that are too long to go into. And thanks, Mike. It was a really, really interesting talk. I have a couple of questions. I was really struck by how vigorous the response of the males were to the funny-looking bright blue bird.
Right.
And I'm wondering if there's any, if you have any information on whether the color per se is what is eliciting responses both in males or females, or whether it is the, I'm not sure how to put it, the intensity of the color, like the intensity of the hue. And I'm wondering if you have any information of how females would respond to those bright blue models, perhaps looking at, lighter blue versus darker blue models. And of course, I'm thinking about the fact that the female's nervous system may be stimulated more by the intensity of the color and not just by the color itself. And then I have the other question is, I think I had to let the dog out. I think I might have missed something, but at some point you talked about brown males that look more like females, and it wasn't clear to me whether that was in the same species or in some other species, because you didn't talk about those. And so I'm wondering whether you have any thought about or any evidence for what's going on with the brown males.
Yeah, so I'll answer your second question first. We've actually done a lot of work looking at brown versus ornament. We call them ornamented versus unornamented. So in this same species that I've been talking about, some males breed in ornamented bright plumage, the red and black plumage that I sort of highlighted in this talk, but other males are in brown plumage. And some of those males in brown, and they look just like females. And some of those males who have female plumage coloration are helpers, they're non-breeding helpers, but others are breeding males. And so we do know that if you're a breeding male, it's better to be ornamented. Your extrapair mating success is much higher. And females have preferences. And we have done, in this case, we've done the mating trials to show that females, if you give a female a choice between a brown male and a ornamented red and black male, the female prefers the red black male. That's consistent with the paternity results we've gotten. And so that's been a whole separate area of research that we've been looking at and asking questions about, well, why would males adopt brown plumage coloration if it leads to lower reproductive success? And I'm happy to delve into that further if people are interested. We've actually published more on that than we have on the speciation stuff that I've been talking about. But the intensity question is a really good one, and it actually goes along with the earlier question about, well, why might females have a preference for redder males? So I gave one hypothesis, which is that maybe it's adaptive. Redder males are, showing that they have good genes, they're in better condition, what have you. So the redness would be a condition-dependent trait. But an alternative is it has something to do with the wiring of the female nervous system, and that just a more intense color triggers a stronger response. And we can't distinguish between those with any of the data we have. The kind of experiment you just suggested is one that never occurred to me. Take a mount or a male that has a totally different plumage coloration, like blue, and do high intensity, low intensity blue, and just see how the birds respond to that. That's a brilliant experiment that I never thought of. I like it a lot, and that might help get at some of these kinds of questions. But we've never done that. Those aren't the kinds of experiments we've done yet. I am getting very interested in the female's perceptive ability and the role that plays. like I was saying earlier about the retinas. But that's just, that's work for the future.
Yeah. Yeah, no, it's really, really interesting. And just a quick follow-up. So the brown males, they're brown for life. They don't.
No, they're not. Yeah, sorry.
Okay.
It's a complicated story. Older males, if they live, they almost all males become ornamented if they live long enough.
Okay.
But it's in the first year of life, males have three options. It could be a non-breeding helper, they could be a brown breeding male, or they can be an ornamented breeding male.
Okay.
And that's the question we've been asking is sort of what induces a male to take
I cannot.
one or the other of those
I cannot
pathways.
call you in 10 minutes.
Yeah.
To summarize a lot of work into a couple of sentences, it looks like what's happening is that males who are in poor condition and are not receiving a lot of social attention during the non-breeding season as they enter into their first breeding season, if they're not receiving attention from females, they are sort of, I don't want to anthropomorphize, but they're basically on the trajectory to be a non-breeder. And so they take on a less costly plumage, the brown cryptic plumage, and then go into the breeding season. But for some of those males, a breeding opportunity arises, like a breeder dies or something like that.
Yeah.
Then they become a breeder. And these birds will move from being a helper to a breeder within a few hours. A breeding opportunity arises, they're moving to the neighboring territory to fill that opportunity within a day. But they can't remolt. They're stuck then with the plumage they have. And we think that's where the brown males are coming from. The brown breeding males are males who were in relatively poor condition, not in a trajectory to be a breeder, but then do become a breeder by happenstance. And they're stuck with a suboptimal plumage, suboptimal in the sense that their reproductive success is lower because they don't sire any extra pair offspring. But what's interesting is there's a lot of behavioral correlates of that. So males who are in that brown breeding mode are really good dads. They feed their young at high rates. They never leave their territory. They mate guard very closely. Whereas ornamented males, even of the same age, do everything the opposite. They leave their territory a lot. They court extrapair females. They don't feed their young very much. So the plumage color is setting the male up for one behavioral syndrome versus another.
Yeah, and it would be interesting to know whether lifetime reproductive success is the same in the ones that start out as non-ornamented versus the ones that start out being ornamented.
Right.
Because it could be that being ornamented gives, a long-term advantage if you're brown the first breeding season, and you're saving energy or whatever. And but then they are able to make that up.
Yeah, and we have the data to do that. We've been talking about it for years. We haven't quite done that analysis, but the paper that I mentioned at the very beginning of the talk just came out this year in AmNat, looks at the female side of that same question, females who are paired with an ornamented male versus paired with a brown male. And it turns out that females who are paired with ornamented males early on in life and more often during their life have much higher reproductive success. Because they, it's a complicated story, but they basically have more offspring, and they have more grand offspring because their sons are preferred as mates in the next generation.
Great. Thanks a lot, Mike. It's really, really interesting.
Yes, thank you.
So I think we have Adriana next. Yes, and then Megan.
Hi, Mike. Great talk. I'm Adriana Maldonado from Universidad del Rosario in Colombia, and I work with social behavior, social structure mostly. And I have two questions. The first one, it's related to the extrapair paternity, and this is mostly curiosity. Is it females seeking the red males, or is it red males going out and courting females? So who's driving this extrapair paternity?
I would say both. So what you see very few extrapair copulations. Evidence from closely related species, other fairywrens, has shown, especially the superb fairywren, work by Andrew Coburn and his group, shows that a lot of the extrapair matings are happening in the darkness before sunrise. and the females are moving to the territory of the male to copulate, rather than vice versa. And we think the same thing may well be happening in ours. But at the same time, males are pouring off their own territory to court females on other territories, and we see that a lot. So males during the daylight will, you see males on somebody else's territory, they're courting the females, they have that elaborate display, they have the flower petals, which I mentioned earlier, but I want to highlight is one of the cutest things a bird can do. I mean, they actually bring fat flowers to the female, but mostly to extrapair females, not to their pair mates. So that's, you can interpret that however you want. But you see that all the time, but you almost never see an extrapair copulation. So we think a lot of courtship is happening and the male's driving that, but the actual decision on whether to mate is sort of up to the female, and she can move to the male's territory if she wants to mate with him pre-dawn. Another really cool result that we've gotten and haven't published is that during these extrapair mating courtship displays that we see, when the male leaves, he usually gets chased off by the territory male or something like that. But when he leaves, he has a very slow, exaggerated flight away from the female, where in another species of fairy wren I used to study, we called it the seahorse flight, because their back is kind of arched and they're fluttering away very slowly, showing their back coloration to the female. But they always fly in the direction of their territory in this very slow, exaggerated way. So I think, one way our hypothesis, which we have not tested, is that they're doing a waggle dance. They're showing the female the direction towards their territory. It's like, come over here before dawn tomorrow, and we can mate.
Some sort of a signal. Oh, that's cool.
Yeah,
So
it's mostly hypothesis and conjecture, but that's what it seems like happening.
Cool experiments that can be done there.
Right.
Very quick follow-up on that. So females engaging in extrapair copulations, do they have higher reproductive success? Is there any benefit? Because we saw the higher reproductive success on males. So from the female perspective, is that also a benefit?
We've compared within pair to extrapair young in terms of condition and survival and things like that. It's a little hard to do because of dispersal. We've not found any obvious pattern. I think we can explain female mating preference for bright males with respect to social pairing because of the results I was just explaining earlier. But for extrapair mating with bright males, we have not detected an obvious or strong benefit at this point. And there are different possibilities. One is it's a subtle effect that's hard to detect, or we just haven't looked at the right thing. It's also possible that the extrapair mating preference kind of spills over from social pair preference. The females just develop this preference for, brighter males. But we don't know. At this point, I can't say how females benefit from extrapair mating.
Oh, cool, interesting. And then my second question that was actually, and this is on the red population. I would assume that there is also a gradient of redness in the males. So my question is,
not a lot. Yeah, there's very little variance in coloration within populations that are outside of the mixing zone. In that mixing zone, the hybrid zone, there's a lot of variation in plumage coloration. Outside of that, relatively little. I mean, there is some, but it's probably not really outside of our measurement error, to measure coloration.
Oh, that's interesting. Okay, because my question was like if they were preferring red, like males that were more red. But if there is not such variation, then what are they looking for?
They certainly prefer males who are ornamented versus unornamented. But in terms of, and I think I would hypothesize or suggest that in the past, when a new allele emerges at some locus that affects male back coloration, that allele can go to fixation. But once that goes to fixation, everybody kind of has the same color. and you don't see a lot of variation. But then some other allele comes in, or a migrant comes in with redder plumage, and that can start the process again. So, that's conjecture. I think that and this is actually consistent with a lot of other studies, like red-winged blackbirds here in North America. Dave Westney looked at this for years, couldn't find any evidence for why females might prefer these extrapair males, and no evidence of effective plumage coloration or anything like that. So I think it's very possible. I would hypothesize that these things go to fixation quickly, and then the female has her preference, and the males are all kind of similar to each other, and then you just wait for another genetic variant to come along. The alternative is that it's very condition dependent, and there is a lot of variation in something like plumage coloration that depends on male condition. But we're just not seeing that in our system. We don't see much variation in back coloration, let alone anything that's condition dependent.
Cool. Thank you, Mike.
Sure,
Awesome.
thank you.
Maren, you're next.
That was a follow-up question to Zuleyma's question. Is there any difference in the survival of brown males, colored males, and females? So natural selection, essentially.
Yeah, no, that's great, and the short answer is no, which was surprising. So we did survival analyses, and we find no difference between helper males, breeding males, or brightly colored males versus cryptically colored males. Females, I think, have slightly lower survival. That probably has something to do not so much with plumage, but with cost of reproduction. But even there, it's not a strong effect. We think, and we actually have a paper that's coming out soon. I think it's just been accepted, or maybe it's still in review, but I think it's been accepted. Joe Welkland, who is another graduate student working on this system, looked at the costs of having bright plumage coloration in these birds. And we don't see any ecological costs of like predation, survival types of things. We think the main cost is social. Males who are brightly colored receive a lot more aggression from other males in the population compared to males who are cryptically colored. Joe did a nice experiment where he manipulated male plumage coloration with hormones, actually made some males molt into brighter coloration, and they were attacked by conspecifics, and including, in one case, a brother who attacked his own brother who was brightly colored and had a vicious fight, and we never saw that male who was attacked again. I don't know if he left the study area or died, but there was a pretty vicious attack there. So I think the main costs of having bright plumage in the system are social, and they're coming from conspecifics rather than predators or something like that. You would still think that should show up as a survival effect, but it could be a pretty subtle one, because males who only the males who are in the best condition or whatever, or who are destined to be a breeder, are the ones who are molting into bright plumage.
Okay, thank you. I will ask a question from the YouTube as well. So it's Nina. She's asked with a follow-up question, and sorry if I miss this info during the talk, but do females who are already paired with red plumage males still go for EPC with red males?
Yes, they do. Yes, yeah. So there's a very high level of extrapair mating. So females who have ornamented mates are still mating with other males, and we don't know why or what the benefits are. Certain, but we definitely know that the males who are siring most of the extrapair young are the ornamented males and not brown.
Thank you.
Do we have any follow-up questions? There's some colleagues who have been silent today. That's okay, but this is your chance. If there's something you want to ask, follow up, or we have some students in the audience. I really want to emphasize that I did notice how much mentoring Mike has done. So many of us are very, very involved in teaching and mentoring If you guys have any questions about that, that's also possible. Well, I'll just share something, Mike. It's for you as much as it can be for anyone in the audience. But because of what we did last week, and really looking at your career in the CV, we have been trying with FINE to produce teaching materials and to get all the research that so many of you are doing on behavioral ecology and social evolution to the classroom. And what I was thinking is if we should try to explore ways in which we work in pairs or with colleagues whose focus is on teaching, as opposed to some of us who are trying, our focus maybe we're doing a lot of research, we're not teaching as much, but there's some of you, some people in the audience that I know are devoted teachers. It would be so fantastic if we can just kind of come up with teams, right, where we do it all together. I don't get to teach a lot animal behavior. I don't get to teach a lot of behavioral ecology. Just don't have the opportunities. There are not enough students, and it's like I'd love to find ways in which I can contribute to the effort of some of my colleagues who are teaching many more students than I am on these fields and topics. Mike, what do you teach? Yeah.
I teach an animal communication course, and then I give lectures in the introductory behavior course, both of which I love. I really enjoy teaching.
How many
but
students,
I think
how many students in the animal communication
course? Well, the intro course has about 180 students probably in it on average. And the communication course varies from 20 to 45 students, maybe, depending on the semester, which is a good number. It's small enough that you can have more interactions. I like that. And it's upper division, so it's graduate students and undergraduates. But I would say, in response to your earlier question, Eduardo, these kinds of things like this, seminars online where people can tune in from various institutions with different kinds of foci from different countries. I think these are wonderful educational opportunities and mentoring opportunities. And I'd love to see more of it. During the pandemic, I think we kind of learned that we can use the tools available to broaden reach and outreach in particular. I happen to be, at the time when the pandemic hit, I was meeting coordinator for our conference coordinator for the American Ornithological Society. And, we went within the space of a couple of months from planning an in-person meeting to a virtual meeting. And we had, I don't know, it was, I think over 2,000 people participated in that meeting because it was online and virtual. It was both a horrible thing that we couldn't have an in-person meeting and a wonderful thing that we could have such broad reach. So I think, FINE and seminars like this are a great model that we should expand on for increasing interactions and reach.
But we're not getting undergrads. And we think we know what their reasons, and I don't think it may not even be reasonable. Since we're not getting as many undergrads as you would like, I'm trying to imagine ways in which we can get to the undergrads. I was listening to you, and again, we get excited with these things. The organizers have a meeting next week to take in so many suggestions, but preparing, if we had modules that some of us who do not get the opportunity to talk to many students, but if we had modules that we make available for colleagues who are teaching every semester courses to 200 students, right? I mean, take sexual selection as a mechanism of speciation, boom. And 1, 2, 3, or 4 of us who have done research on that, I have not, say, listen, if this is a lecture that would help you in your course, here it is. It's ready. We can offer it virtually to make it more personal. We can visit the course, a couple of us for a few minutes.
Yeah.
So bring it to the classroom as opposed to we're not getting the track that I would like to get of the students coming here. Yeah.
Reaching undergraduates is always a challenge, whether it's virtual or in person.
Yeah.
But I do think that there is some model there that you're sort of talking about that could work. The challenge, the biggest hurdle is to get them, I don't know, motivated enough that they want to participate, despite the fact they have a lot of other competing demands on their time. That's what I always find the biggest challenge with undergraduates, is they have a lot of competing demands on their time.
Adriana is right. Let me just thank the community, our colleagues who join us via YouTube. So this is a good time for me to stop the live streaming. So if you're watching us via YouTube, please remember you're always welcome to join via Zoom. There's no password code, there's no password. Just show up, use the link.